Replication of DNA. What happens during DNA replication? How is a new polynucleotide strand formed? Why is the process of DNA replication called semi-conservative? So just to see where this fits in the biological molecules topic. So we're here, nucleic acids are important information carrying molecules, and it's DNA replication. I'm looking at the specification reference from AQA. The semi-conservative replication of DNA ensures genetic continuity between generations of cells. The process of semi-conservative replication of DNA in terms of unwinding of the double helix, breakage of hydrogen bonds between complementary bases in the polynucleotide strand, the role of DNA helicase in unwinding DNA and breaking its hydrogen bonds, attraction of new DNA nucleotides to exposed bases on template strands and base pairing, the role of DNA polymerase in the condensation reaction that joins adjacent nucleotides. Students should be able to evaluate the work of scientists in validating the Watson-Crick model of DNA replication. Replication of DNA. So cells are derived from existing cells by division, and there are two stages. There is nuclear division, which is where the nucleus divides, and cell division, where the whole cell divides. Before the nucleus divides, the DNA must be replicated to ensure all daughter cells have genetic information to produce enzymes and proteins. DNA replication is precise so that new cells are identical to the original, and it is known as semi-conservative replication. So, the first thing that happens is the enzyme DNA helicase breaks the hydrogen bonds between the bases on the two polynucleotide strands, and this makes the helix unwind to form two single strands. Then, each original single strand acts as a template for the new strand. Complementary base pairing means that free-floating DNA nucleotides are attracted to their complementary exposed bases on each original template strand. So A pairs with T and C pairs with G. So the bases match up using complementary base pairing. Then, a condensation reaction joins the nucleotides of the new strand together. It is catalyzed by the enzyme DNA polymerase. Hydrogen bonds form between the bases on the original and the new strand. DNA polymerase joins the nucleotides via a condensation reaction, and the hydrogen bonds form between the strands. Each new DNA molecule contains one strand from the original DNA molecule and one new strand. We've got one original and one new strand. One original, one new strand. DNA polymerase action. So each end of a DNA strand is slightly different. One end is called the 3' end, and the other is called the 5' end. During DNA replication, the active site of the DNA polymerase is only complementary to the 3' end of the newly forming DNA strand. So the enzyme can only add nucleotides to the new strand at the 3' end. So the new strand is made in a 5' to 3' direction. And DNA polymerase moves down the template strand in a 3' to 5' direction. DNA polymerase action. Because the strands in the double helix are anti-parallel, the DNA polymerase working on one of the template strands moves in the opposite direction to the DNA polymerase working in the other template strand. Semi-conservative replication needs 4 nucleotides. You need adenine, guanine, cytosine and thymine. You need both strands of the DNA to act as a template for the attachment of these nucleotides. You need the DNA enzyme DNA polymerase, which catalyzes the reaction, and you need energy to drive the process. So, enzyme DNA helicase breaks the hydrogen bonds linking the base pairs of DNA. The double helix separates into two strands and unwinds. Each exposed polynucleotide strand acts as a template to which complementary base pairs are attracted. Energy is used to activate these nucleotides. Activated nucleotides join together by enzyme DNA polymerase to form missing polynucleotide strands on each of the two original polynucleotide strands. Each new DNA molecule contains one original DNA strand, half the original DNA saved and build into each new DNA molecule. So it's semi-conservative replication. So look at a worked example question now from AQA. So the diagram shows part of a DNA molecule. How many nucleotides are shown in the diagram above? Remember that one of these is a nucleotide. So it would be 8. Name the type of bond labelled X in the diagram. Well, you're making the sugar phosphate backbone, and you're joining here, so it's a phosphodiester bond. The enzyme DNA helicase and DNA polymerase are involved in DNA replication. Describe the function of each of these enzymes, DNA helicase and DNA polymerase. The DNA helicase unwinds DNA and breaks the hydrogen bonds, and DNA polymerase joins nucleotides or forms phosphodiester bonds. So, the semi-conservative replication of DNA ensures genetic continuity between generations of cells. The process of semi-conservative replication of DNA in terms of unwinding of the double helix, breakage of hydrogen bonds between complementary bases in the polynucleotide strands, the role of DNA helicase in unwinding DNA and breaking its hydrogen bonds, attraction of new DNA nucleotides to exposed bases on the template strands and base pairing, the role of DNA polymerase in the condensation reaction that joins adjacent nucleotides.